A good day again. In lecture 17, we talked about the prokaryotic cells. We said that these are the cells which don't have a what? They don't have a nucleus, and we say that they don't have organelles bound membrane like this. These organelles which have those membranes surrounding them. This is what we talked about in lecture 17. So now we are going to go deeper into the examples of these cells. So, microorganisms. Microorganisms play a very important role in our lives. They have a positive impact and they have a negative impact. So one of the negative impacts of microorganisms, we say that they can cause diseases. Many of the diseases which are there, they are caused by microorganisms, and we say that they are, but majority of them are harmless because we can't live without microorganisms. They also have that vital part in that they play in our bodies. Some of them they are used in digestion, some of them they are used to carry out some processes, and even to provide or to aid in the oxygen that we are breathing. So, what are microorganisms? What are microorganisms? So, they are very small living organisms. So small that most of them they are invisible in the using our naked eye, meaning that we can't see microorganisms unless we use other special equipment to see them. Unless we use other special equipment to do what, to see them. So they can only be seen using a microscope most of the time, and even using a microscope you can't see all of them. There are other special microscopes which are some of them they can be seen using a microscope. So, but they make up 60% of the earth living matter, these microorganisms. So estimated that there are close to 2 to 3 billion microbial species, and which share a planet with us. I'm so sorry for the disturbance which is taking place. I'm just recording this video as for an agency. I really know that we'll be writing mid June exams, and we didn't cover a lot of theory, so I had to do this via. And my voice is too low because I'm doing this in an office. So a lot of people, a lot of people are getting inside the office, and others people are busy there and there. So I don't want to disturb them, but I hope you can hear what I'm saying. So, classification. We can say this. They have been classified according to their smaller kingdoms, because at the end they differ, these microorganisms. Others are called bacteria, that are under the kingdom Monera. Others are called protista, under the kingdom Protistia. Others are algae. Algae is according to taxonomy. It depends. Some they can put it under kingdom Protista, or in Bio 112 they will explain it under the plant section, but not saying that they are plantaya or kingdom Protista. But according to other sources, they just classify them as separately. Then, so fungi is under the kingdom of fungi. So we also have viruses. These are the 5 microorganisms that you are having: bacteria, protista, algae, fungi, and virus. But in this, we are going to go deeper into bacteria, then we go deeper into viruses in this bio triple one. So note that only members of the kingdom Animalia and most Plantae are not considered microbes. Why? Because they have what you call eukaryotic cells. Actually, we said that we first learned about prokaryotic cells. These are cells without the nucleus, or without the knot or the kernel. So now these cells are Animalia in the Plantae. They are under in classifications, or let me say that they are now cells. They are now cells. So viruses are also considered microbes, but viruses are not cells. Why? When research why viruses are not cells, and under this group under this link, video link, you just you explain your answer or mark there, or you message me on WhatsApp why viruses are not cells. Each kingdom has its own associated viruses. Each kingdom has own associated viruses. Many of us know bacteria only as germs, invisible creatures that can invade our bodies and make us sick, because from junior school they talk about bacteria. But size of a typical cell compared to a typical bacteria. A typical animal and plant cell is 10 is to 100 micrometers, whereas a bacterium is less than that. So it's 2 meters of diameter, 2 micrometers of diameter. That's the size of a bacteria. So imagine if it's 2 micrometers of micro. Micrometers, it means 10 negative 6. 2 times 10 negative 6 meters. It's too small. You see that it's too small, small. So morphology. What is morphology? Morphology is classification based on shape and size. Morphology is classification based on shape and size. So bacteria occur in different shapes. We have what we call rods or bacilli. These are singular bacillus. That's form of a rod. Others they are classified under spheres or cocci. They are these are like spheres, like with a sphere. Do you know an egg? So they're like eggs. They're like eggs like this. Others that are called they're in spiral form. So the spirida bacteria, spiria. No, just take a pencil and draw a line like the zigzag line. So that's a spiral. So the other bacteria like that. So I'm going to classify them like that. Others are called filamentous forms. They grow in branching, so to form like filaments like this. So the interchanging filaments. So interchanging filaments, just take two pencil and just draw lines like this. So these are just the filamentous forms of bacteria. So we classify like that. So in the test, they're going to demand you to know the rods, example of a rod, example of a sphere, a spiral forms, and filamentous forms. But these are the examples that you need to know. Like they're so simple. So we can see the classification of bacteria there. You can see a cocci, a sphere. You see a sphere. You can see also a bacilli there. You can see in others that are spirida. Like the name of bacteria. There's a bacteria called streptococcus. You can see that it ends with the cocci. That means that it ends with a form of. You can use the names to classify them. Other names, look at the names which that contain the bacillic form, like bacillic name is term inside, as others are containing a cocci term inside. So that will be a cocci related the bacteria, and others like that which have the bacilli there. These are the bacillus bacteria. So how can we name bacteria? So we use what you call the linear system to name bacteria. So we name the genus, and then we name the species of the bacteria. So the Escherichia coli is named after the Theodor Escherichia, who discovered the organism in 1885. So other bacteria we can name using the scientists who discovered such bacteria, like Theodor Escherichia discovered this Escherichia coli in 1885. But we can have the other bacteria, Bacillus megaterium. Bacillus megaterium is a large rod-shaped organism, a member of a genus Bacillia. You can see that it is Bacillus megaterium. So from the name Bacillus megaterium there, you can see that there is bacillus. That means this is a rod-shaped organism, megaterium. So you can see that you can use the name of bacteria there to discover its morphology. Well, is it a rod? Is it a rod? Or is it a sphere? Is it a filament? We can just use such. So the name often reveals some characteristic feature in them. So importance of microorganisms. So they form diseases. So they They cause diseases. Most infectious diseases are controlled by proper sanitation, but they cause diseases. This we know this is a negative impact to us. So agriculture, they are vital in terms of processing. Some they fix nitrogen inside the soil. Some even in manufacturing we can find this in like agricultural based manufacturing in making oil, gas, and all this kind of stuff. So food and drinks, I talked about it, about bread. You need microorganisms in this alcohol beverage, in wine making, in also making of other traditional alcohol, chibuku. You need also these microbes. So chemical products, in other pharmaceuticals and antibiotics, you need what you need these antibodies. Even some import to culture other hormones, you need these and microorganisms. So they are so important. So they are also used in biotechnology, in the ability to move genes freely from one to home, like genetic engineering, part of biotechnology. We use these microorganisms. So they are used in basic research. Why? Because they grow faster. Within 20 minutes you can have millions of bacteria, and you use that to test, to conduct researches. Imagine if you use eukaryotes, they grow slowly. Yeah, so you better test some other things and other drugs into them, so that you can see how because that the first cells, that the simplest cells, so it means they're simpler to study. After all, if they have simple processes that you can lead us to what we need in researches, so we use them and conduct this basic research. So in bioremediation, breaking down sewage and other toxic waste into safe matter. Sometimes we don't know where to throw these sewages, so we just take them into the sewage, and because what we want is that byproduct. So something interesting about this microorganism, that byproducts is what we need. Some they produce free they produce environmentally friendly products that we may we obtain and we use. So, in other ways, we can say that these microscopic organisms, they play a key role in maintaining life on Earth. They are fixing gases. They break down dead plant into let the plant and animals matter, so that decomposition, and also biotechnologists exploit the activity of microbes to benefit humans. Because in other words, we can't live without microorganisms. That would be a lie. We can't live without them. So that will be the end of my lesson.